Hey guys, welcome to part 2 of the video on genetic technology, and in this video we will talk about gel electrophoresis, and this is mentioned in the syllabus that we need to know how to analyze proteins and nucleic acids via this technique, and also how to distinguish between alleles of the same gene using gel electrophoresis. So you really need to get an understanding of the principles behind this process. So it's basically a technique used to separate different molecules, and we use it extensively in analysis of proteins and DNA. And what is the basis of the technique? Well, it involves placing a mixture of molecules into wells that are cut into agarose gel, and then an electric field is applied, and the movement of charged molecules within the gel, because the gel has pores, in response to the electric field depends on a number of factors. Number one, what the net charge of the molecules are. The negative molecules will move towards the anode, and the positive will move towards the cathode, and the ones that have a greater charge will move faster because the electric force of attraction will be greater. For those of you who are familiar with Coulomb's law, this will make sense. And size is also a determining factor, because the molecules that are smaller will move more quickly than larger molecules. So a smaller DNA fragment will move more quickly than a larger DNA fragment. And the third is actually not a property of the material that is being separated, but rather the composition of the gel which is being used, because agarose gel is one possibility. There are other types which can be used. For example, polyacrylamide for proteins and agarose for DNA, and it is actually the size of the pores within the gel through which the molecules move which determines the speed with which the proteins and the fragments of the DNA will move. So three factors you need to keep in mind: that both the charge and the mass matters when it comes to separation via gel electrophoresis. And obviously the type of gel that is used is also a factor. So we can carry out electrophoresis of both proteins as well as nucleic acids. So let's study about proteins first. So what does the charge on a protein depend on? Well, it depends on the ionization of the R groups on the amino acid residues, and you know there are many different kinds of R groups. So if you have an NH3 on an R group and the material that the molecule is in happens to be alkaline, then that particular residue will not be ionized, will not carry a charge. But if it is in a pH of 7 or in an acidic pH, then the amino acid residue will carry a charge, will be ionized. And those of you who are studying chemistry at A levels will really find it easy to grasp this concept. So you will remember from chapter 2 that some amino acids have R groups that can be positively charged. My apologies, I was saying NH3, but what I meant was NH2. And some have R groups that can be negatively charged, that is COO minus. And whether they are charged or not is dependent on the pH. When proteins are separated by electrophoresis, the procedure is carried out at a constant pH using a buffer. And why do we want a constant pH? Because we want the conditions to be the same. then all of them will move towards the positive end. So interesting. This is interesting because gel electrophoresis can be used to separate the polypeptides produced by different alleles of the same genes. So they will be similar, but they will be slightly different because they will be different in terms of either charges or perhaps different in terms of length. For example, allozymes are variant forms of enzymes produced by different alleles of the same gene. So, adult hemoglobin is composed of 4 polypeptides, 2 alpha globin and 2 beta globin. And in sickle cell anemia, a variant of beta globin has an amino acid with a non-polar R group instead of one with an R group that is charged. So this variant basically has the same chain length, but its charge is different. How so? Because it has a non-polar R group instead of a polar R group, it will have slightly less charge and so will move a smaller distance. And the variants can be separated by electrophoresis because they have different net charges. So this means that hemoglobin molecules in people who have sickle cell anemia have a slightly lower negative charge than normal hemoglobin, and so the molecules will not go as far through the gel as a molecule of normal hemoglobin. So to test whether or not someone carries the sickle cell allele can make use of this difference in the net charge, because we just said that proteins have an overall negative charge, and because of the absence of the R group, the variant will have a slightly less negative charge. We can write this down. Sickle cell hemoglobin has a slightly lower negative charge. So won't move as far up. That is towards the anode. And we have figure 19.9. Where is it? Which shows the separation of different forms of hemoglobin in samples of blood taken from members of a family which has a child with sickle cell anemia. Damn it. So you must realize one thing, that before electrophoresis can take place in DNA, you need to actually fragment, that is to separate the fragments. But in the case of protein gel electrophoresis, it is considerably more easier because we don't have two strands of the same DNA, so things are easier. So this is separation of hemoglobin by gel electrophoresis carried out on a family in which one child has sickle cell anemia. Lane 1 contains hemoglobin standards, right? A is normal hemoglobin, NS is sickle cell hemoglobin, right? Lanes 2 and 3 are the samples from the parents, and 4, 5 and 6 are from their children. So 3 contains, both 2 and 3 contains one band at A and one band at S. And as you know, sickle cell carriers are, are expressing both the normal gene and the abnormal gene. So some of their hemoglobin will be normal and some of the hemoglobin will not be normal, and that's why we will have both bands, right? But 4 is not a carrier and so only has a band over here. 5 is also a carrier because it has both bands. However, 6 is not a carrier, it is actually someone with sickle cell, and that means that none of the hemoglobin that he or she produces is normal, and so there is no band over here because both of the alleles are abnormal. A carrier has a normal and an abnormal, and a normal person just has the normal alleles. Both of his alleles are identical and normal. So electrophoresis of DNA. And that's it. So DNA fragments carry a small charge thanks to what? In proteins it was due to the ionized R groups, but in DNA it is due to negatively charged phosphate groups. Yay to phosphate groups. And so both proteins and DNA are negative, which is an interesting thing to note. And in DNA electrophoresis, the fragments will move through the gel and towards the anode, and the smaller the fragment, the faster they move. So in this scenario, charge isn't going to have an impact, only the size matters. We look at an example, the use of genetic profiling in forensic science. Figure 19.1 shows how genetic profiling is carried out. Regions contain are something called VNTRs, which are variable number of repeated DNA sequences. So at that position place, most people would have the same repeat sequences, but the number would vary between people. So if there was a repeat sequence GAC, the people would have this sequence at the same spot, but just how many repeats of GAC the person has would be different. Obviously, people, even those who are not related, can have the same number of GAC at a point, but it is unlikely that at the VNTR sequence at in another part of their body, in another part of their chromosome would also be the same. So that is why they compare VNTR sequences for a lot of different places in the chromosome. And only identical twins share all of their VNTR sequences. You can share quite a few of your VNTR sequences with your parents, and even with in a non-related person, some of your VNTR sequences might be the same, but it is impossible that all of your VNTR sequences are the same, except for your identical twin. DNA can be extracted from almost anything that has come from a person's body, the root of a hair, tiny spot of blood, etc. And usually the quantity of the DNA is increased by using PCR, which makes many copies of the DNA that has been found. And you really need to get an understanding of PCIs because it has been specifically mentioned in the syllabus that you need to know the principles of PCR and the involvement of cloning and application of DNA with reference to the Taq polymerase. So you have the DNA. So a detective found some hair at the scene of a crime, right? And all of the DNA was extracted from the cells of that hair. Now we have the DNA. That is one huge extensive DNA. Now what does the forensic scientist do? The forensic specialist do? Well, the DNA is first chopped into pieces using restriction enzymes. So really, restriction enzymes are really important. They come into play here as well. And they use those restriction enzymes that are known to cleave it close to the VNTR regions. And that is why the fact that restriction enzymes are so specific comes really helpful. And if you find this a bit difficult to understand, think of it this way. So we have gene A, and then we have VNTR sequence, and then we have gene B. And if we were to compare hundreds of people, this would be the same. They would have gene A, then they would have repeat sequences of G, C, A, and they would have gene B. And this would be the same in all of them. The difference would be in just how many GCA repeats there are. Someone may have 10, 15, 30, 40, whatever. But because the A and the B are the same in everyone, so it's easy to cut close to the VNTR region because just about at the end of the A, there may be a specific target site for a particular restriction enzyme, and just near the end of B, there would be another specific restriction site for perhaps the same or another restriction site. And that is how this particular VNTR would be separate. There will be complementary strands, and that is why heating of the paper will be required. And then short sequences of single stranded DNA called probes are added, right? And they will have base sequences that are complementary to different VNTR regions. And the probes contain a radioactive phosphorus isotope. So when the paper is placed on an X-ray film, the radiation emitted by the probes, which are stuck to the DNA fragments, will make the film go dark. So we end up with a pattern of dark stripes on the film, which will match the position that the DNA fragments reached on the agarose shell. Alternatively, the probes may be labeled with a fluorescent stain that shows up when ultraviolet light is shown onto them. So this is actually really interesting. Maybe I should repeat it because they often ask questions about this. So DNA fragments, they carry a small charge due to what? Due to a negatively charged phosphorus group. And if you were to take a look at the syllabus, they say you have to be able to distinguish between alleles of a gene, right? So if a person is heterozygous at a particular gene locus, meaning he may have a normal hemoglobin allele and an abnormal hemoglobin allele, it means that he or she will have two bands instead of just one. There's a person who has both, who is homozygote for the normal hemoglobin allele would have just one position, right? Something to remember. And if they ask you the question why all of the fragment move towards the anode, you will have to tell they do that because all of the DNA is negatively charged and so will naturally be attracted towards the positive electrode. And if another question is the distance moved by the DNA fragments, what factor does the distance moved by the DNA fragments depend on? You will say it is dependent on the size of the fragment. Or more likely, the mass of it. So different sizes will cover different distances. So if you were to compare two alleles or two of the variants of the beta polypeptide chain,